Hi everybody, welcome back. Miss Calabrese here. So in this video, we're going to be talking about antimicrobial medications. Right, so first, a little bit of terminology to get us all on the same page when we're talking about these drugs. Um, First term up here, chemotherapeutic drug, that's going to refer to any chemical that we use for treating pretty much anything. So a lot of times we think of chemotherapy in reference to anti-cancer drugs, but technically chemotherapy is any kind of drug that we use to treat or relieve pain or for prophylaxis. Prophylaxis technically refers to using a drug for preventative reasons, so to prevent infection of some sort. And then antimicrobial chemotherapy, that's when we're specifically using drugs to fight infection, to fight some sort of microbial disease. Right? And so antimicrobials is something that we use for any microbial drug, no matter what it does or where it came from, which is going to be different from the term antibiotic. So antibiotic refers to an antimicrobial that has a natural origin, right? So that means that it comes straight from nature as a natural origin for that particular drug. Semi-synthetic drugs are ones that are taken from a natural origin, but then are adjusted, right? So that we make some chemical modifications to make them stronger or more broad spectrum or something, so that they're more effective than the natural form of that compound. Synthetic drugs are those that are created entirely in a laboratory that have no natural origin. And then the terms broad and narrow spectrum refer to how widely these particular drugs can be used among microbes. So if they can be used among multiple different types of microbes, then we would refer to them as broad spectrum. If they can only be used in a limited number of species, then we would refer to that as narrow spectrum. Okay. A little bit more terminology here about the combinations of antimicrobial drugs. So a lot of times drugs are not prescribed all by themselves, but they're prescribed in combinations with other drugs. So if we're prescribing drugs and one enhances the effect of the other, then we would refer to that as synergistic. So one drug kind of helps the other one be even more powerful. If one drug interferes with another drug, then that would be an example of an antagonistic relationship. Um, and if we've got drugs that have two separate effects that neither are synergistic nor antagonistic, then we refer to that as additive. All right, so mechanisms of drug action. So these are the basic categories by which most antimicrobial drugs work. A lot of drugs work by inhibiting synthesis of the cell wall. So if you could prevent a microbe, a bacterium, from creating more peptidoglycan to reinforce his cell wall, then eventually he'll die. Some drugs work by inhibiting the synthesis of nucleic acids, so stop you from making RNA, stop you from making DNA, and that's going to prevent the cell from functioning. Inhibition of protein synthesis, as you can imagine, is going to involve interfering with the ribosome. So if we can't make proteins at the ribosome, then we're not going to be able to survive. Interference with cell membrane structure and function, and then inhibition of folic acid synthesis. So this is like influencing a metabolic pathway that's important for maintaining life. All right, so here are those main categories again. So, and some examples of drugs that fall under those categories. So as far as cell wall inhibition, those are beta-lactam drugs like penicillins and cephalosporins. As far as plasma membrane inhibition, there aren't too many of these and they're kind of narrow spectrum, but this would be things that are like polymyxins. Things that inhibit protein synthesis are going to interfere with ribosome activity. Those would be tetracyclines, aminoglycosides, macrolides. Metabolic pathway, folic acid inhibitors would be like the sulfa drugs, sulfonamides. And then synthesis of nucleic acids like DNA and RNA, interfering with those would be things like rifamycin and the fluoroquinolones. All right, so we'll talk about some of these main categories and a few of the most common drugs in those categories. So as far as the cell wall blockers, so the drugs that inhibit creation of peptidoglycan. Those would be penicillin, cephalosporin, are common drugs in that category. A lot of times they're referred to as beta-lactam drugs, and that's because of we're referring to the chemical structure of the drug itself. So beta-lactam drugs inhibit the cell wall. All right, so penicillins are natural form of this drug, so an actual antibiotic, meaning it has a natural origin. And the origin of penicillin is mold, a mold of the genus Penicillium. So these drugs are most effective against gram-positive cocci. So that's what the GPC stands for there. And that's because if you can kind of visualize this the cell wall of a gram-negative, they've got uh, um, they've got an outer membrane outside of the cell wall, and that outer membrane is going to prevent the penicillin from actually getting to the cell wall itself. So these are most effective against gram-positives. There are some semi-synthetic versions of penicillin where they've been kind of augmented to make them more powerful, so that they can infect, or they can affect gram-negatives. And those would be things like the ampicillins and amoxicillins. So these drugs have a very high level of selective toxicity. And by selective toxicity, I mean they're going to be toxic to the microbe and are very rarely toxic to humans. And that's because the mechanism of action is to target peptidoglycan, which is not something that humans have. So really the only downside is that because these originate from mold, and mold is a common allergen, that these can cause allergic reactions in some people. Right. So making penicillin more effective, since penicillin was the earliest antibiotic, bacteria have had a really long time to evolve resistance to it. And one of the things that bacteria do to resist penicillin is they develop an enzyme called beta-lactamase. And this beta-lactamase actually destroys the beta-lactam ring of the penicillin. So one thing that we can do is add an ingredient that will destroy that beta-lactamase enzyme, right? So, and that would be things like in the antibiotic augmentin, we have amoxicillin, which is the beta-lactam drug, and then we add this clavulanic acid, which is going to inhibit the beta-lactamase so that the bacteria are no longer resistant to it. All right, cephalosporins. So these are another cell wall inhibitors. They're very broad spectrum. They tend to have less allergies associated with them, fewer allergies than penicillins. All right, and then a handful of other drugs here that inhibit the cell wall that you might be familiar with. So aztreonam is good for people that have penicillin allergies. Bazotracin is a good one for topical application. So surface like skin infections, you would use this, but you would not take it internally. And then vancomycin for people that have infections that are, for example, like methicillin resistant infections, you would take vancomycin. All right, moving on to our next category of antibiotics, here are the protein synthesis inhibitors. So here we've got a picture of a prokaryotic ribosome. And so just to refresh your memory, prokaryotic ribosomes are smaller than eukaryotic ribosomes. So they've got their 70S ribosomes, which means they're composed of a 30S and a 50S subunit. And those subunits are slightly different than the subunits of a eukaryotic ribosome, which means that we can design drugs that are going to inhibit this ribosome, but are not going to impact your own human ribosomes that you have inside your cells. Right, so some main drugs that fall into this category are the aminoglycosides, tetracyclines, macrolides. Right, so starting with the aminoglycosides, these guys bind to the small subunit of the ribosome, and basically by binding there, they're going to prevent protein synthesis. So they kind of get in the way of the ribosome's job of being able to read messenger RNA and create polypeptides. These drugs don't typically work well against anaerobic bacteria, but they work fairly well against aerobic bacteria. Now, a lot of times aminoglycosides are given in combination with beta-lactam drugs, and that's so along with a penicillin of some sort. And that's because a lot of times the aminoglycosides are going to have a difficult time gaining entry into the bacterial cell so they can find their way to the ribosome. But administering them along with a beta-lactam drug will allow the beta-lactam drug to weaken the cell wall, which is going to allow the aminoglycoside entry into the cell. Okay, so why are aminoglycosides administered along with beta-lactam drugs? Give you a second to think about it. I'm not trying to be tricky here, but there is a difference between A and B. All right, so the answer is going to be B. So the beta-lactams are going to make the cell wall weak. They're not going to have any effect on the cell membrane, so only the cell wall. All right, so tetracyclines are another type of protein synthesis inhibitor. So these guys also bind to the small subunit of the ribosome. These are very, very broad spectrum drugs to the point where that's not just bacteria. They work on some protozoans too. They can have toxic effects on a lot of different cells. Tetracycline's very broad spectrum protein synthesis inhibitor. All right, and then macrolides. So macrolides are going to bind to the large subunit of the ribosome. They're mostly effective against gram-positives, gram-positive drugs, and these are used commonly for people with allergens to penicillin. So these ones you've probably heard of erythromycin or azithromycin or clarithromycin. So any of those myosin drugs are macrolides that are going to be targeting the large subunit of the prokaryotic ribosome. Right, and then chloramphenicol, this is another large subunit protein synthesis inhibitor. This one is not used too often. It's more of a drug of last resort because it does have this potentially lethal side effect of aplastic anemia. All right, so moving on to the cell membrane inhibitor. So now we're talking about the phospholipid bilayer, things that can interact with that. And this again, these tend to be pretty narrow spectrum drugs. So polymyxins are an example of cell membrane inhibitors because they interfere with cell membrane function. Right, so, and polymyxin is a natural antibiotic. So this comes from, they're actually derived from bacteria in the genus Bacillus. So colistin is an example of this one. And they disrupt the cell membrane. They're going to be pretty narrow spectrum and again, usually only effective on gram-negative rods. One benefit of this is that it can treat some drug-resistant strains of Pseudomonas. Negative is that they tend to be toxic to the kidneys. And moving on to the nucleic acid synthesis inhibitors. So these are the ones that are going to interfere with DNA replication, interfere with transcription. The fluoroquinolones are a common one. They inhibit the unwinding of DNA. So remember that that in order to replicate our DNA, we need to kind of unwind it. And so the fluoroquinolones interrupt that process of unwinding. So if we can't unwind it, then we can't do replication of DNA, and we also can't do transcription. So that's how fluoroquinolones work. Rifamycin works by inhibiting transcription exclusively by stopping the active enzyme RNA polymerase. Okay, so yeah, these fluoroquinolones, this is a totally synthetic drug. They prevent the unwinding of the DNA. So Cipro would be an example of this one. They are broad spectrum and very strong drugs, but there are some negative side effects, especially as you get older, more likelihood of developing seizures, and this one, which always creeps me out, the possibility of tendon rupture, even months after taking a fluoroquinolone antibiotic. Okay, rifamycins. So rifamycins are the ones that are going to block RNA polymerase, so they're going to stop transcription. So these ones could be relatively broad spectrum as well, so effective against a lot of gram-positive and some gram-negatives. So these are used to treat things like tuberculosis, to treat leprosy or Hansen's disease, and again, relatively broad spectrum. All right, and then our last category of antibiotic drugs here are the ones that inhibit metabolic pathways. Here we're mostly talking about inhibition of the ability to create folic acid. So these are folate inhibitors, which means they don't allow the bacteria to make its own folic acid, which is something that bacteria need, just like you need folic acid, except we get folic acid from our diet. Bacteria have to synthesize their own. So if we can stop the metabolic process of folic acid synthesis, then we can prevent them living, essentially. So sulfa drugs are one of the larger examples of this. Trimethoprim works a little bit further down the metabolic pathway. Right, so these sulfonamides or sulfa drugs, totally synthetic, and they work by competitive inhibition. So when we talked about enzymes, I believe last week or the week before, we talked about how we can interrupt enzyme function by acting as competitive inhibitors, so creating a molecular mimic that's going to insert itself into the active site of an enzyme and prevent that enzyme from catalyzing any further reactions. All right, and then trimethoprim works a little bit further down that metabolic pathway, but still is going to inhibit the production of folic acid. All right, so those were the major categories of antibacterial drugs, so the ones that are targeted against bacteria. So now we're going to move on to just a quick mention of antifungal drugs. So one thing I want to point out is that fungus, they are eukaryotic cells, right? So whether we're talking about a yeast infection or a mold infection, um, these are eukaryotes, not prokaryotes. So the cell structure is going to be a little bit different, which means that our cellular targets are going to be a little bit different, right? So if we want to say, for example, target the cell wall of a fungus, that cell wall is not made of peptidoglycans. We're not going to be able to use the same drugs. Most fungal cell walls are made of chitin, sometimes cellulose. Um, but we can still target things like cell membrane. We can target nucleic acid synthesis. But it's the selective toxicity for antifungal drugs is not as good as the selective toxicity for antibiotics for bacteria. And that's because fungal cells they're more like our cells. So it's more difficult to find these pathways that are going to be exclusively toxic to the microbe, but not toxic to the human host. Right, one of the common types of antifungals is this one, ergosterol, where we're targeting ergosterol. So ergosterol is sort of like cholesterol. So you have cholesterol inside your cell membranes to help strengthen them. Fungi, instead of having cholesterol, they use ergosterol. So if we can inhibit that, we can weaken the cell membrane. So a lot of these, like amphotericin B, a lot of these azoles, like fluconazole and imidazole, those ones are inhibiting ergosterol. Right, and then as far as drugs that are anti-protozoal or anti-helminthic, unfortunately, we don't have a whole lot of drugs that fall under these categories, just because as we get to the protozoans and the helminths, now we're talking about cells that are very, very similar to our cells, right? So protozoans are eukaryotic cells that are very similar to the way our cells operate. And then helminth cells, they are animal cells, right? Helminths are animals, so their cells are basically exactly like our cells. So of the drugs that work on protozoans and helminths, a lot of them are ones that are going to be interfering with metabolic pathways. Some of them are just straight up paralyzing agents. So for a lot of the worm infections, you take a drug that essentially paralyzes the worm's muscles, so that he can no longer adhere to the inside of the gut, and he'll pass out with the feces. Okay, moving on to viruses. So viruses, remember, are going to be different from all of these other categories because they're not considered living cells, right? So we can't have a cell membrane inhibitor because there is no cell membrane. We can't have a cell wall inhibitor because there is no cell wall. So we have to work with what viruses give us to work with. So we have to think about the viral mechanisms of reproduction, which is that lytic cycle, if you can remember back to. So attachment, penetration, biosynthesis, assembly, release. If we can interrupt that pathway, that makes a good antiviral drug. So Most of the antiviral drugs are either going to try to inhibit viral entry, so stop that virus from getting into the host cell, stop the uncoating process, so stop the part where we take off the envelope and the capsid, stop the nucleic acid synthesis, or stop the assembly and release of viral particles as the major categories of antiviral drugs. And we don't have a lot of good antiviral drugs out there. And it's because of the way viruses attack, because they're inside your own cells. It can be very difficult to attack viruses without doing a lot of damage to your own cells. Right, so as far as entry inhibitors, so these are the ones that stop viruses from getting in. Uh, Entry inhibitors are going to prevent that virus from docking with its spikes onto the host and prevent the endocytosis that brings the virus in. Ones that stop the uncoating of the virus, so at this point the virus is already entered into the cell, but we're not going to unwrap that package. We're going to leave it wrapped up in its envelope and capsid. And then the most common type are the nucleic acid synthesis inhibitors. So these are by far the most common type of antiviral drug, where we stop them from reproducing, stop them from basically hijacking the nucleus to make more of its own nucleic acids. Okay, so quick question here. Would you be able to look at these major categories of antibiotics and pair them with how they work? So penicillins, cephalosporins, fluoroquinolones, aminoglycosides, polymyxins, sulfonamides. Would you be able to match those with these mechanisms of attack? Right, so here's your answer. So penicillin and cephalosporins, those are both cell wall inhibitors. So they inhibit the synthesis of new peptidoglycan. Fluoroquinolones inhibit nucleic acid function, right, because they prevent the unwinding of DNA. Aminoglycosides inhibit protein synthesis by adhering to the ribosome. Polymyxins interfere with cell membrane structure, so they're interfering with the phospholipid bilayer. And then sulfonamides are going to inhibit metabolic processes, so specifically the production of folic acid. All right, I hope that was helpful. Let me know if you have any questions.